episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor.
Welcome, everybody, again to another episode of our show with another truly fascinating guest who is helping to create a better tomorrow for all of us. Today, we have the honor of being joined by Dr. Nora Khaldi, who is the founder and chief scientific officer of Nuritas, which is a company that's helping to solve some really extreme challenges in both biology and medicine, and is reconnecting to a principle we talked a little bit about on previous shows, basically as foods as the best medicines with some really interesting cutting-edge 21st century technologies. Dr. Khaldi has a PhD in molecular evolution and bioinformatics from Trinity College in Dublin, a master's in mathematics from Aix-Marseille University. And she's in the process of really disrupting the status quo when it comes to drug discovery by integrating tools like artificial intelligence and big data to extract and isolate some really novel bioactive peptides from food-based materials for the development of therapies to treat a wide range of health disorders. Since launching in 2014, Nuritas has really grown rapidly, has a wide range of well-known funders, including support from EU Horizon 2020, the European Investment Bank. They've raised over $65 million to date. In 2017, Dr. Khaldi was named Woman of the Decade in Business and Leadership at the Women Economic Forum European Union event, received the Rising Star Prize from Tech Excellence Awards. In 2019, she was inducted into the Young Global Leaders, the World Economic Forum's Foundation for Remarkable Leaders Under 40. And she continues to lead the Nuritas team, ultimately in developing products for billions of people with life-changing ingredients and ultimately medicines, working with a variety of global leaders like the Nestle company, BASF. Really exciting stuff. Dr. Nora Khaldi, thank you for taking the time to come on the show and talk to us for a little while.
Thank you. Thanks very much, Aaron. Really nice to be here. I've watched you interview incredible people, so I'm happy to be here.
It's great. It's great to have you. Typically we love to start off by handing the floor back to our guests for a little bit just to talk about themselves. If you could take us to start on just a little journey of your path, obviously in mathematics, how you moved into bioinformatics, molecular evolution, and also, if you wouldn't mind, if you could touch on, I know it's not going to be the topic of this show, but a little of your early work in the area of horizontal gene transfer, because I think this is a very important sort of milestone in your earlier research that I think we should all know about as well.
Cool, thank you. So it's been an interesting journey, Ira. So I was always attracted to science, and like very early on, since I remember, really interested in science, constantly questioning things. All my heroes were scientists. I remember this is going back years ago when I was maybe something like 7 in class, and the teacher said, okay, what do you want to be? And a friend of mine just turned to me, and we still talk about it today. And she said, okay, what do you want to be in your future? And I said, I want to be a scientist. And she's like, oh, okay, what is that? And I started explaining, you discover things and you help people. And I think that's really the journey. I was always attracted to life science and science in general. So then during my career, what happened, it was interesting. At the end of high school, I was really good in both areas, so biology and mathematics, pure mathematics, and or mathematics in general. And I remember saying, okay, and those disciplines when you go to university are totally separate. And it's a pity, but at that time, totally separate, even separate universities. So I just said, okay, so which one do I prefer? And it was a really hard decision. And I remember talking to different people and so forth. And then finally, I decided to go with pure mathematics because I just loved it. I really enjoyed it. So I went with pure mathematics. And I remember going through the years and as you know, pure mathematics is very theoretical. Most of it is not connected to any reality. And I remember once I got my master's degree, I was walking on the street one day and I met a doctor. She's my family's doctor, and And she said to me, okay, how are you getting on? I said, yeah, I just got my master's. Oh, okay. And what? Pure mathematics. Oh, great. And what do you do with pure mathematics? You know, she's like, what do you do? Do you want to become a teacher? And I'm like, no, there's so much you can do with pure mathematics. And it's funny because people don't know the extent of what can be done with pure mathematics. You can literally go into anything. And I was always, like I said, attracted to biology. So that's the second part of my career when I decided to continue into a PhD, but more application of the mathematical part, the computational part I had into biology. And the area that intersected between both was bioinformatics. So I did a PhD in bioinformatics and molecular evolution. And really the whole PhD was about understanding these molecules, these mycotoxins that were produced by species around us to compete against other species. So it was an arms race development of these active compounds that a lot of them actually turned into become really interesting drugs. Penicillin is one of them, etc.
Sure.
And my goal was, how do we computationally improve the discovery of these molecules? How do we find them faster? These are very complex molecules sometimes. Why do these species express them? How do they exchange them? Do they exchange them? And this is where the whole horizontal gene transfer came about. So it's an observation I had made when looking at these incredible networks of genes or clusters of genes that were giving rise to these incredible molecules. These genes were all, you know, in the genome together working as a factory to create one molecule that the species expressed to maybe kill off other species and to outcompete other species. And it was interesting to see how these genes came together. Very often you'd see them cluster in evolution through millions of years of evolution in these species. It was incredible. But then one of the observations I had made was that was the link between these different clusters in totally different species. And at that time, a lot of horizontal gene transfer was known in eukaryotic species, in unicellular species only. And then what I discovered was that pluricellular species could actually also have gene transfer. And this is where the whole discovery came about, that we could transfer genes between different species. And that then, as a consequence, in terms of phylogenetic trees, etc., there's a consequence on that, because obviously phylogenetic trees are based on the fact that only very simple species like bacteria could exchange, but not more complex species. So it put into question a lot of the phylogenies that were built as well. So that was an interesting discovery. I made quite a lot of discoveries, created a lot of software programs that are still used currently by industry and academia in terms of identifying those clusters, identifying the compounds different species make. It was an interesting area, really interesting area. And like I said, a lot of these were to discover drug precursors. So it's kind of a drug discovery part to it as well.
Sure.
And then what happened after Ira is that I was really interested in continuing. I had, created an interesting background in that area. People recognized my name. I had, quite a lot of publications and really interesting ones. But then I got approached by a consortium that was coming together in Ireland, and they were interested in looking at dairy and looking at a totally different area and understanding whether there was components in dairy that could improve human health for various reasons, for improving their value, for improving human health, etc. And I hesitated because I didn't really know this area. Never, my understanding of food was like everyone else. You take food, you gain energy, and that's it. Some food is good for you, some food is bad for you. But I didn't know the extent of what you could find in food. And so I took the position, and it was really looking at the computational part of their discovery. So they were using traditional ways of discovery, which is basically random screening.
Sure.
Like everyone else, and it was all random screening, and it wasn't going really anywhere in terms of it. It's very expensive, it's very tedious, etc. A lot of work went into it. They found some interesting things, but it was taking too long. It was too expensive. So the goal for me was, is there another way around this? And coming from my math background, I realized that the biggest issue in this area is really down to simple data. There's too much data, molecular data to test, especially from nature. It's full of molecular data. So how do you identify the right compounds on computers before you enter the lab so that you're selectively only taking the best ones forward?
Mm-hmm.
And this is how I started working in this field. My first realization was that we're not using food to its full potential at all. That there's so many compounds out there in everyday food and in nature in general that we aren't even aware of, and they are so potent and do various interesting things from a human perspective, and that there was no other way to access them without bringing in new technologies. The traditional ways of screening and researching was never going to work. We had to apply complete new thinking to this area. And this is how I started really developing this whole new thinking. And Nuritas came at the end of that. A few years later, realized that this is not just a problem of, this consortium. This is a bigger problem of the whole industry. And consumers are looking for more understanding of what they're taking, and also trust in whatever, if they want to consume something, that it's actually helping them. They're looking to improve their health span, live healthier lives for longer, etc. And then food companies and all the industry in itself are also looking to differentiate and to tackle those issues. But there are no solutions. There are no really good solutions, or very few good solutions that have really true science behind, that can really be used in a personalized way to tackle a lot of these issues.
It's a really exciting domain. I had sort of in a previous career some exposure to it, and some of the things that you're bringing up again, obviously sort of looking at the 20th century, and you mentioned penicillin, of course, but we have a very large pharmaceutical industry that was basically created by natural products, but a lot of the smart people have, always said, well, we only scratched the surface of even nature, let's say. You're talking about sort of even deeper than that. Beyond the small molecules, you have all these peptides and other bioactive moieties. It's really infinite, hence the tools. I took a little, ride through some of your publications today. Really fascinating things that span from, looking at rice for novel inhibitors of inflammatory cytokines. You have a fascinating project with fava beans and muscle atrophy, antimicrobial peptides from peas. Obviously, you can create a company here that goes in many directions. Just talk, if you would, about some of these lead projects, if you would. Obviously, nothing confidential, but that sort of give us a sort of very interesting profile here on just common foods in these cases that can really do really interesting new things for us.
Yeah, absolutely. So maybe before I start, one thing I think that people don't realize is that, and I say this in my talks usually, but I usually show an apple, and I always say that within that apple, there's more molecular data than all social media put together since the inception of social media. Yep. So we know very, very little of what we do every single day, which is eat. And it's crazy because it's the one thing we can alter to improve our health. Yep. And to prevent disease, and it's not used. We're not using it the right way at all. So the question from a Nuritas perspective was, how do we define better understanding of what's out there? Mm-hmm. Also define certain actives within those products that have a specialized function that improves a special thing in humans. So I'm going to give you some examples. So we went after then, the technology is a platform, so literally can be pointed towards any consumer need. Mm. We went after different consumer needs like muscle health in general, which is basically muscle retention. It's one of the biggest indicator of aging. It's the first thing you see physically when you start aging. You sag, basically, you drop in muscle, but it has huge consequences in everything. They've connected it now even to different neuronal problems, including Alzheimer's, etc. They've connected it to immunity issues. They've connected, obviously mobility is a big one, but loss of muscle is a big indicator of age. How do we actually halt it or maintain muscle for longer? And there are currently no solutions. Yes, there's whey, some HMB, etc. But those are more nutrients when your cell is working healthy. But when you want to, shift your cell from where it is to a healthier state, so that it consumes the nutrients better, that's what's needed, a solution to maintain muscle for longer. So we went after the muscle arena. We went after metabolism as a second one. Metabolism, I think, is a big one. As we age as well, in general, we don't metabolize as well. And especially glucose metabolism is a big one. So I'll tell you a little bit more about that area. But we went after pre-diabetics and our people with high glucose levels in blood, and seeing whether we could reduce significantly that. And then separately, we went after things like anti-inflammatories, etc. And the real core behind it is that at the end of the day, what we want to improve is human health. And we're targeting areas that we believe current solutions are not performing the way they should be. Our current solutions are not answering the question. And that nature has provided solutions to these, it's just that we haven't found them. So for the muscle one, for example, it's a network of peptides that we selected from fava bean. And this network, we've done preclinical work now. It's currently in 2 clinicals, and we're starting a 3rd one. And we've launched a product, with the Peptides Strong. It's called, the product is called Peptides Strong already. And this one is really interesting because it has 3 mechanisms of actions that are totally different, and it brings together 3 mechanisms that are important for muscle. One is reduction of TNF alpha, specifically inflammation. So reduction of inflammation. One, it reduces the genes that are responsible for muscle breakdown. So it reduces a bit their expression. It just tampers it a little, and it enhances the synthesis of muscle, more protein. And together by those 3 different mechanisms, so it's not a 1 mechanism approach, it's a 3 mechanism approach, all found in fava bean. Now, when you eat the fava bean, you don't get the benefit. It's a pity. Maybe some might get it because they might digest very differently the fava bean to others. But in general, if you look population-wide, you don't get the benefit. It's only really by unlocking these molecules called peptide from the protein structure that you do get this activity. So we unlock them, we find them, then we unlock them, and we in a very natural way, and then we move it forward. And it's really interesting to see the effects of this that we're seeing at the moment in terms of reduction of muscle loss, reduction of inflammation, and the cells, you can see them even under a microscope becoming healthier, so metabolizing better, etc. So I think that one is a really interesting one that we're moving forward with. The other one is in the metabolism arena. The metabolism is really important. We went after, like I said, pre-diabetics, but we could literally go after any person that just wants to, have a normal level of blood glucose in their blood so that they don't develop any further issues in the future. And there are many studies where they looked at, population-wide studies where they looked at thousands of people and found that even if you drop your glycated hemoglobin, which is the indicator of glucose over time, HbA1c, by just 0.1 to 0.2%, you can reduce mortality or augment your health span by 10 years. You can reduce mortality by a massive number, I can't even remember, 25% or something. You reduce your risk of getting so many different diseases just by that amount during your lifetime. So that was our goal, and we found a network of peptides in P that actually can do that, and it targets. What it does is that it targets muscle cells, and because that's the major powerhouse of the human body, and that's where a lot of the glucose goes in. But when your muscle gets tired and ages, you don't pull in the sugar as well. So that's where we improved that. So glucose metabolism within the skeletal muscle cell. And then the other ones are aging, where we looked at skin aging, etc. Inflammation is another one where we've developed one from rice. And there's like 6 clinicals now. Only 2 are published, but the other ones are on skin, on hair, in different formulations. And the efficacy is incredible in terms of, there's 4 peptides that are important here, all different anti-inflammatories, naturally from rice. And that's the types of ingredients that we're developing. And what's interesting, the reason we went with peptides, okay, there are multiple different, molecules. We went with peptides for multiple reasons. One is that peptides are the major signaling molecule in the human body. So most signaling in our bodies, including in the bodies of animals as well and plants, is actually through peptides. It's the most natural way to signal and to start a conversation with your body and to get your cells moving towards certain directions and shifting certain pathways from one to another. So that's really peptides, and that's the reason we went with them. We also went with them because they're hard coded in the genome. So it's it makes us identify them on and on and on the same way all the time. It can be very different for a small molecule. It really depends. That the expression of small molecule comes up and down. It's very hard to find them. A lot of them may have, side effects, toxicity, etc. But these are hard coded. They're always there. It's all about finding them. And also, they're still considered nutrients. So proteins are still considered nutrients, which is beneficial from an ingredient perspective as well, and a claims perspective. So I think, and then maybe I'll just add on. So those are just the top of the iceberg of what we found. So, we've only developed 3 ourselves, and we're co-developing some with different companies as well behind the scene, addressing very large needs. But really, we have about 3 million peptides behind the scene that are ready to go in development. We've characterized them, we know what they are, we know what they do. And that's just the tippy top of the iceberg. There's so much out there that we haven't discovered that can really alter human health significantly.
It's really exciting, and I think one thing I want to emphasize, because I'll make sure the audience appreciates it, because you mentioned networks of peptides, and I think this is sort of an overlooked thing nowadays, that that fava bean or that pea, it's not making that one compound. Like when I take my penicillin, I get better. They're making these cocktails of bioactive substances. Evolution has done this for a reason. Along those lines, I noticed another interesting paper, and I don't know if you could talk about this one just for a couple of minutes, but you were doing some comparisons between the peptides that are found in breast milk and infant formula, which is, very, obviously a very hot area. Can you just talk a little bit? I don't know if this is confidential, don't worry about it, but can you talk a little bit about the work you're doing here? Yeah,
it's interesting, actually. I think infant formula is a very interesting industry, and when you want to make sure that the baby is having or is getting whatever is needed. If you can't breastfeed them, okay, that the formula milk really represents as closely possible the breast milk itself. So what we did was simply looking, okay, peptides, like I said, are the major signaling molecules in the human body. So milk carries a lot of peptides to the baby, and the reasons are various, from modulating the immune system of the baby, to modulating neuronal development, to modulating the gut microbiome, to killing certain bacteria and promoting others, etc. So peptides have a huge role to play. But a lot of the emphasis on developing formula milk has always been on the proportions of the proteins. So obviously cow's milk is different to human milk. So they played around in terms of, well, let's create proportions of the major proteins that look more like human than cow. And that's fine. They also played a lot with oligosaccharides and additional sugars, basically specific sugars that would feed certain types of bacteria that are important for babies. Super research, incredible research there. But from a peptide perspective, we looked at it and we took quite a few different samples of formula milk, and we compared them to, we looked at that and we compared them to breast milk. So we took samples of breast milk too. And we looked at a comparison. We found that there's very little overlap. That from a peptide perspective, really formula milk is not really representative of human milk at all. And that's an issue in itself. So the question was, okay, well, and it's because those regions where the peptides were coming from were absent in bovine, but present in human. And that's why we could find them in human breast milk, but we couldn't find them in bovine. And the question then was, okay, well, how can we enrich formula milk with the right peptides that look like bovine milk? Obviously, we're not going to go and make them synthetically. So are there plants or other sources around that would have similar peptides to the ones in human, but not in bovine? And we've done a little bit of that work, which is behind the scene. But the first work that you've read was simply saying, and it was a little controversial because no one looks at that area. But, you know, peptides are, you know, the paper, it took a while to get accepted because we got a lot of reviewers saying, no, you know, debating, but here are the results. You know, we're clearly seeing that that there's a massive difference. We separately we randomized. We just simply randomized what we call a hydrolysis, which kind of represents the breast milk and the plant and the bovine milk. And actually, the randomized version was as close to human milk than the formula milk to human milk from a peptide perspective. So that was an interesting project, actually. And it opens up doors in terms of, well, that needs to be addressed.
Yeah, it's a fascinating market in its own right. I haven't had too much exposure to it, but once again, as you were mentioning, this unique cocktail that's in breast milk that does so many things. It's just another nice example.
Ira, that's actually one of the reasons I'm in this area. I went, before I accepted the role in the consortium that I worked with, I went to a conference I didn't know anything about food or milk or anything, but I went to a conference in the US, and this professor from Melbourne was presenting his work on Wallaby Milk. And I was blown away by Wallaby Milk. And I don't know if anyone, your listeners, knows about Wallaby
I didn't even know it existed. I tried cow's milk one time.
There's small quantities, but very relevant. So they did a research in the 70s, and they repeated it multiple times after, where they took wallabies, they come out as fetuses, they're marsupials, and they develop in the pouch. And the only thing that develops them, the only thing they have an interaction with is the breast. So they feed off a breast, and the mom can have multiple nipples feeding different babies at different ages. So what they did was they reversed the milk. So they took the milk of an older wallaby and they fed it to a fetus. And then the other fetus, they just fed them the normal fetus milk, okay? And what they showed was incredible. I was blown away. They showed two images of totally two different wallabies. One was like tiny, skinny, couldn't barely move, no fur, nothing, no muscle. The other one was massive, furry, really robust, hopping. And they're the same age. Exact same age wallaby. The only difference was the milk they were giving. So later milk was developing specifically the muscle and the outside, not the inside. The earlier milk was specifically developing the intestines, the brain, everything that's inside, but not the outside. So when they obviously looked into them further, they found that, one was developed well outside, and that's the later milk, because the milk changes through lactation and sends in specific molecules, a lot are peptides that really just focus on the development of intestines, brain, etc. Next stage now is the outside, but it was incredible. I just the power of coming out of there. I'm like, okay, if one were to understand what's in those, what's in that milk and how it works, it's just incredible.
Wallaby milk. Who'd have thought?
Yeah, wallaby milk. Yeah.
We'll talk about that separately.
Yes.
Nora, one of the, obviously you had a tremendous amount of success on the science and this clinical development. You've also been very active, as I mentioned in the intro, with working with some of the global leaders, Nestle, BASF. And one of the things, I guess, just to people not realize that, unlike, say, the drug industry where we're developing that drug and it's going to be the drug with the dose and all that, when it comes to consumer products and foods, a lot of other things come into play. This peptide in this milk product, let's say, it better taste good or I'm not going to drink it. Dr. Nora Khaldi might drink one of these things a day, but Ira Pastor might drink 6 bottles of it. So talk a little bit about how some of the sort of the anecdotes, let's say, about working with the food and consumer products industries for these novel products, if you would.
I think it's a very good question, Ira. So basically, I think you touched on 2 things. One is the consumption at the end, but also I think the first part is really the development. So it's good to find something in nature that has a health benefit, but to take it from that discovery all the way to a finished product actually is there's a lot.
Yeah.
Okay, there's a lot. It has to taste good. You have to be able first to make it. Okay, so first step is, can we make it naturally? Okay, or do we have to go synthetic? But can we make it naturally? That's our question. Can we make it naturally? Mm-hmm. If the answer is yes, then it goes to the next step. Okay, so can we make it naturally in a cost-effective way? Because if it's not cost-effective, people aren't going to buy let's say we put it in a drink and the drink currently costs $2. They're not going to pay $5 for the same drink or $60 for the same drink, even if it gave them wings. It's like, okay, how do you create a cost-effective solution? Yeah. And then the second thing is, does it taste good? Because if it alters taste and it tastes terrible, again, the adoption of it is going to be low. Yes, you can still launch it, but the adoption is low. And then also within the formulation that you're going to put it in, is it free to act, or is it like, do you lose it through the production of the products? And there are so many things. And then also, things like, well, when I eat it, does it break down? Because a lot of actives just get broken down in the digestive system, so you never really get their efficacy. So that's important. You need to show, okay, well, our active doesn't. Does it reach the destination it needs to reach to act and to signal, and all that's important. And so all those different parts of the puzzle we had to build from scratch, and it took a while to build because, but what we did, which was interesting, was actually part of that was to develop AI and machine learning predictions at each level. So for every one of those, we created data behind the scenes so that we can predict at the very beginning. Instead of just predicting for efficacy, we'd also predict for, is it going to be stable? Can we scale it naturally? Can we actually extract it? Is there enough available, or is it too low that extraction is not going to work? Is it going to work in humans? So is it orally available? And so those are questions we build predictors for, to reduce the risk of us taking something forward through development and then it failing along the way, because you want to reduce that risk. Absolutely. So that's one way, and we've built that system, and we've done that now multiple times, and we've learned throughout. We've done our mistakes as well, where we've moved with certain things and we realize actually, dosing is actually too high here. We need to bring it down and so forth, because of cost, simply because of cost. But we've done that multiple times now, and it's great to be able to say we could literally point the technology now to any health area, and we could develop, products in about a 2-year cycle, which is really interesting. In this area, and then compared to like the 30-year to 100-year cycle, which is the traditional way, which is crazy. So that's good. But separately, also with food companies, the goal here is to say, okay, so what's interesting, and this is a consequence of pulling it out of nature. We're not saying, for example, the fava bean product, we're not saying it's equivalent to the dose of our ingredient is equivalent to eating a kilo of fava bean. It's not. It literally is nearly equivalent to eating the same quantity of fava bean. But maybe a 10% increase on top of what you're eating from a fava bean. And that's important because, you know, you don't want to say, okay, it's equivalent to, you know, eating 10 kilos of fava bean. And if you eat 3 of those at 30 kilos of fava bean, that's not the way so it has to be in a proportion that makes sense.
Sure.
And then the question that we got at the very beginning as well, for example, the glucose regulation one is like, well, if I eat so many, does that mean my glucose is going to drop? And the funny thing is that no, and that's the interesting about peptides. Peptides only perform when you need it.
Yeah. So
when your glucose that day is healthy, it's just nutrients. When your glucose is high, that's when it performs. And that's interesting. And I think that's why naturally made ingredients that have gone through the whole hundreds of years of evolution are really smart that way.
Absolutely.
It's very hard to mimic that by design.
Yeah.
You know,
nature is good at doing that.
Nature is incredible. That's why we just, you know, I got a lot of questions before, why don't you just design? And we could design easily. There's nothing better design than what's out there.
Sure. Yeah. Complete agreement on that.
Yeah.
Nora, you know, I've watched several of your presentations prior to talking, and one thing you talk a lot about, I've seen you talk about, is the sort of balancing this dual role. You're a scientist, but you're also an entrepreneur now. As one presentation was like, you know, if anyone tells you can't do both, they're nuts. I usually get a lot of questions from sort of the newer generation that's coming along, that may be starting out. Give some messages, if you would, to the folks that are just getting started and maybe doing some exciting stuff in the lab, but haven't taken that step yet. And please.
Okay, so targeted towards scientists then.
Yeah, or vice versa. Yeah, well, yeah.
Yeah, my own background. I think there's a big question you have to come to in your career in science, and I think that's for me was the determinant moment. It was, do I want to work and create publications that maybe one day will make it through to something that would help an individual at the end? Or do I want to actually go faster and help the individual today? Well, by today, it's relative. It's not today, but it's relative compared to a publication. So, and that was a deciding moment in my career where, you know, I had seen 2 versions of it. I had seen people that have just went after the publication, which is great. I played that too during my PhD, etc. But very rarely is someone else going to pick up that publication and push it forward to make it happen from a consumer perspective and really helping an end consumer. So I was always, you know, I loved science because of that angle of I want to help consumers during my own lifespan. You know, I want to help them during my own career.
Yep. And
that's why I decided to not only stay in university and continue the research I was doing, but actually take it out and focus on getting it to consumer faster. And it's a big decision you have to make. It's like, okay, do you want to just, and that's fine. Both areas are absolutely interesting. They both need a lot of work. There's a lot of pushing in both areas. There's a lot of work. There's sleepless nights. But in the area of, you know, developing something the whole way, there's a lot more learning you have to go through. So, you know, publication, there's a way, academia, there's a certain things you need to learn. When you're taking something from pure research into something in the lab all the way through to commercial and then talking to companies and commercializing it, there's a lot of different steps that are totally different. That are, you know, a lot. You have to learn a lot during that. So I would say, you know, both areas would need a lot of work, dedication, trust in your own capability, a a lot of, you'll always get a lot of people around you saying, well, this is impossible. This is impossible. No one has done this before. This is impossible, too expensive to do. They tried before. Why you, it literally is, there's a lot of negativity. Just remove it. Keep moving forward. It's very hard to create new. It really is. It's very hard to create something that isn't there, because you have to create the journey. Sometimes you have to actually lay it out, and that's much harder than just saying, I'm a me too, but I'm better. That's an easier game, but when it's a me one, it's always much harder, but you make it work. And then, so for me, it was really trying to get to an end product and a person faster than through publication.
Outstanding, and a great message to everyone that's going to be listening. Nora, it's been a fascinating journey so far. I'm really wishing you the best with all this. The portfolio is fascinating and developing on so many fronts. It's going to be really fun watching the continued development. For everybody that's going to be listening to this episode on the podcast or watching on the YouTube channel, you've been listening to Dr. Nora Khaldi, founder and chief scientific officer of Nuritas, going after some extreme current challenges and unmet medical needs in medicine, reconnecting us with this fascinating principle as foods as our best medicines, and ultimately connecting it with some really cutting edge 21st century technologies and approaches. Nora, it's been a great time talking to you. I really want to thank you for taking the time out of your schedule to do this. Thank you for the work that you've been doing. And as we say to our guests, thank you for helping to create a better tomorrow through your research and through, in your case, your entrepreneurship as well. It was really a great time listening to it.
Thanks very much, Ira. Thanks for having me. It was really nice. Thank you.